Where is DNA found in eukaryotic cells? Of course, the nuclear DNA is found in the nucleus. This is the total of the autosomes and the sex chromosomes that we've been discussing. But there's also DNA in chloroplasts and in mitochondria. In most cases, mitochondrial DNA and chloroplast DNA is inherited through the maternal line. Egg cells are much larger than sperm cells. In most organisms, sperm convey only a haploid set of nuclear DNA. The egg provides the cytoplasm, the organelles, and all the rest of the cellular machinery for the next generation. The mitochondria in your body are clones of the mitochondria from your mom and her mom and down through the generations. The only genetic variability in mitochondria comes from mutations during DNA replication. Therefore, comparing mitochondrial DNA is a great way to track maternal lineages. An interesting application of this is from a study documenting the skeletal remains of what is thought to be King Richard III. King Richard III died in August of 1485 and was the last king of medieval England. Richard was defeated and killed in the Battle of Bosworth. Richard III was childless, and the kingship was already in dispute, so hence the battles. His body was unceremoniously dumped in an unknown location until 2012, when an archaeology team was able to extract a skeleton of a 32-year-old male with scoliosis. Richard III has been portrayed as a monstrous hunchback, but his disability was actually not as terribly pronounced and was still capable of fighting in battles. Radiocarbon dating confirmed that the remains to be over 500 years old, matching up with the timeline of Richard's death. DNA analysis using mitochondrial DNA confirmed the skeletal remains match the mitochondrial DNA of two known female line descendants of Richard III's sister, Anne of York. Because the mitochondrial DNA of both living descendants and the skeletal remains and all the other lines of evidence, researchers are highly confident that the skeleton remains are indeed that of Richard III. The interesting part comes from the Y chromosome, the chromosome that is inherited through the paternal line. Remember that sex chromosomes don't usually undergo crossing over, so the Y chromosomes passed from father to son also typically do not have much variability, which is only due to mutation. Because Richard III did not have any sons himself, the male lineage was tracked through the descendants of his two brothers. This resulted in a 19-step comparing of lineages from father to son. At some point, there was at least one false or misattributed paternity, which is when the father on record does not match the biological father. At this point, the researchers cannot determine at which point in these 19 steps the misattributed paternity or paternities could have occurred. But the Y chromosome from the skeletal remains of Richard III does not match the Y chromosomes in the verified living descendants. This study is a great example of how mitochondrial DNA from maternal lines and the Y chromosome from paternal lines can be used to determine the identity of a missing person, the heritage of someone, and a number of other interesting applications. Tracking the paternity in plants is a little bit harder. Most plants are hermaphroditic. Conveniently, pines inherit their chloroplasts through the paternal line. Pinyon pines, or Pinus edulis, at Sunset Crater near Flagstaff, Arizona, show two different kinds of growth forms, and for the sake of today, we'll call them the normal growth and the wonky growth. The wonky trees are pruned by a moth that, as caterpillars, bore into the growing tips of the branches and kill them. The normal trees are resistant to the moths, but the wonky trees are susceptible. We suspect that the moth resistance or susceptibility is a heritable trait. The interesting thing about these trees in the context of today's conversation is that the wonky trees rarely produce female cones. In most conifers, female cones are produced near the tops of the trees, but the wonky trees have functionally had their tops removed by the moths. The question that the researchers set out to answer is, to what degree do the wonky trees contribute to the gene pool of the pinyon pines of the Sunset Crater population? Researchers led by Kitty Gehring, my graduate school advisor, determined that about 80% of the seeds they collected share the same chloroplast DNA from wonky trees. So the wonky trees were started to be called superdads. These two studies show how non-nuclear DNA can be used to understand inheritance.